everyone, welcome back to the Pharmacy India. In this class, we are going to discuss top 25 MCQs questions of biochemistry, which is a very important subject. And these questions will be very important for your upcoming Telangana Pharmacist exam. This is your mock paper 5. And before starting to the question, make sure to download Pharmacy India mobile app from the Google Play Store to get latest pharmacist updates, and you can also join Pharmacy India's other social platforms like WhatsApp and Telegram, Instagram to get latest pharmacist updates. So let's get started with our first question. The first question is oxidative phosphorylation occurs in. Now you have to tell the place where oxidative phosphorylation takes place. So the correct option will be inner membranal surface of cristae. What is cristae? Cristae is the innermost area of mitochondria. Mitochondria. Let's draw a diagram of mitochondria. Basically, mitochondria is a double membrane bound cell organelle. Double membrane bound sausage shaped cell organelle like this. This area, inner membrane, this area which inner membrane forms, it is known as cristae. Cristae. And the inner side of this cristae, inner membrane of this cristae, the oxidative phosphorylation takes place here. Alright. So the correct option will be inner membranal surface of cristae. Next question, all of the following statement about the nucleus are true except. Now you have to choose the correct option which is absolutely true about nucleus. The first option is outer nuclear membrane is connected to endoplasmic reticulum, that is ER. This is absolutely correct. Next, it is the site of storage of genetic material. I think the question is wrong. Question should be nucleus are true. Okay, you have to tell the option which is not true regarding nucleus. Okay, next is it is the site of storage of genetic material. It is also a correct option. Next, nucleolus is surrounded by a bilayer membrane. No, nucleolus is not surrounded by a bilayer membrane. So this will be your correct option. Next is outer and inner membranes of nucleus are connected at nuclear pores. This is also a correct statement about nucleus. But third option, which is nucleolus is surrounded by a bilayer membrane. This is not true regarding your nucleus. So the correct option will be third. Next question, von Gog's disease is characterized by the deficiency of the enzyme. Basically, von Gogh disease is a glycogen storage disease. Glycogen storage disease. Your options are glucokinase, glucose 6-phosphatase, phosphoglucomutase or glycogen synthase. Basically, glucose 6-phosphatase, it is an enzyme whose deficiency causes von Gogh disease which affects your liver and muscles. Affects liver and muscles. Primarily liver and also muscles. So the correct option should be glucose 6-phosphatase. Clear? Next question. The rate limiting enzyme in glycolysis is. Now, from the given options, you have to choose that correct option which is a rate limiting enzyme in glycolysis process. Now, what is glycolysis? Glycolysis is a type of cellular respiration, or you can say metabolic process. Metabolic process in which glucose gets converted into pyruvic acid. Pyruvic acid. It is a 10-step process. 10-step process. Clear? So your options are phosphofructokinase, glucose 6-dehydrogenase, glucokinase or pyruvate kinase. Basically, these all are the enzymes of glycolysis. But from the given options, phosphofructokinase is also known as rate limiting enzyme. Clear? Rate limiting enzyme. So the correct option will be A, phosphofructokinase. Next question, phenylketonuria result due to the absence of the enzyme. Now question is asking about that enzyme which is deficient in the disease phenylketonuria. That is also known as PKU. Options phenylalanine oxidase, phenylalanine hydroxylase, phenylalanine transaminase, or phenylalanine oxygenase. Basically, phenylalanine hydroxylase will be correct option. Now, what is phenylketonuria? See, your body contains phenylalanine, which is a metabolic essential amino acid, you can say. Phenylalanine gets converted into tyrosine with the help of this enzyme, which is called phenylalanine hydroxylase. But for example, if phenylalanine hydroxylase gets deficient or becomes less, or the amount of phenylalanine hydroxylase decreases, phenylalanine gets accumulated in some parts of your body and causes phenylketonuria. So the correct option will be phenylalanine hydroxylase. Next question, which of the following participate in the formation of coils and helix structure? Covalent linkage, peptide linkage, intermolecular hydrogen bonding, or intramolecular hydrogen bonding. Basically, coil and helical structure, they are the types of secondary structure of protein. Secondary level of organization of protein. Level of organization of protein. Clear? What are coils and helix? Basically, this is helical structure. This is helical structure, and this is beta pleated sheets. Basically, these are the types of secondary level of organization of protein. So, covalent linkage, peptide linkage, and intermolecular hydrogen bonding. These are the wrong options because the correct option is intramolecular hydrogen bonding. Now, what is intramolecular hydrogen bonding? That type of hydrogen bonding which forms between a single molecule. Clear? Between a single molecule. And what is intermolecular? That type of hydrogen bonding which forms between two different molecules. Clear? Correct option will be intramolecular hydrogen bonding. Next question, a positive ninhydrin reaction is due to. Now, the question is from qualitative test of proteins. Qualitative test of protein. Your options are reducing sugar, cholesterol, alpha amino acids, or triglyceride. Basically, triglyceride is a type of lipid. Clear? So this will be wrong answer. Next, alpha amino acid will be correct option because ninhydrin test gives positive reaction for alpha amino acid. Clear? The influence of ninhydrin test is alpha amino acids. So the correct option will be alpha amino acid. Next, move on to the next question. Which one of the following is not an essential amino acid? Now our body contains total 20 amino acids from which 10 are essential and 10 are non-essential. 20 amino acids, 10 essential and 10 non-essential. So from these 10 essential amino acid, you have to choose that amino acid which is non-essential: tyrosine, tryptophan, threonine, or phenylalanine. Basically, there is a trick to learn a total essential amino acid, that is PVT, TIM, HALL. That is private Tim Hall. P for phenylalanine, V for valine, T for threonine, next T for tyrosine, then isoleucine, methionine, histidine, arginine, leucine, and lysine. But tryptophan is a type of non-essential amino acid. Non-essential amino acid. So the correct option will be tryptophan. Now, what is non-essential amino acid? Those amino acids which forms inside our body, and essential amino acids are those amino acids which you have to take through your diet. Okay. Next question. Which of the following amino acid cannot undergo transamination? Lysine, alanine, aspartic acid, or glutamic acid. What is transamination? Basically, just read the term carefully. Transamination. Trans means transfer. Amination means amino group. amino group. So basically, when you transfer an amino group from an amino acid to a keto group, that process is known as transamination. But question is asking, like which amino acid cannot undergo transamination? So from the given option, lysine is that amino acid which does not undergoes transamination. Along with lysine, proline and threonine. There are also the amino acid which does not undergoes transamination. Clear? So the correct option will be lysine. Just mark it. Next question. A fatty acid which is not synthesized in the body and has to be supplied in the diet is. Now you have to tell the example of essential fatty acid. Essential fatty acid. What are essential fatty acids? Those fatty acids which cannot be synthesized in the body and has to be supplied in the diet. Clear? So your options are palmitic acid, lauric acid, linolenic acid or palmitoleic acid. So the correct option will be linolenic acid. It is very obvious. Palmitic, lauric and palmitoleic acid, these are non-essential. 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 And this is also a non-essential fatty acid. Clear? Similar to amino acid. Clear. Next question. The major fuel for the brain after prolonged starvation is glucose, fatty acid, ketone bodies or glycerol. So basically the question is asking like from which of the following molecule or structure your body gets fueled after a long prolonged starvation. Glucose, fatty acid, ketone bodies or glycerol. Basically glucose, fatty acid and glycerol, they are not a type of major fuel for the brain after prolonged starvation. So the correct option will be ketone bodies. Basically our body contain three types of ketone bodies which stores and synthesize in liver with the process of ketogenesis, and also the precursor of ketone bodies is acetyl coenzyme A. All right, it occurs in liver. So the next question is which of the following vitamin would most likely become deficient in a person who developed a completely vegetarian lifestyle? That is vitamin C, means ascorbic acid, niacin, cobalamin or vitamin E. You can see the correct option will be cobalamin, that is vitamin B12. What is the reason? What is the reason behind it? Because vitamin B12 does not have any type of plant-based sources. Does not have plant-based sources. Clear? So if a person who developed a completely vegetarian lifestyle has most likely become deficient with vitamin B12, that is cyanocobalamin or cobalamin, which is the most complex vitamin which contains cobalt or 63 carbons number. So next question is hypercholesterolemia is not found in hyperthyroidism, nephrotic syndrome, hypothyroidism or obstructive jaundice. What is hypercholesterolemia? Basically, if the cholesterol level becomes elevated from the normal range, that is known as hypercholesterolemia, because hyper means high level and cholesterol means cholesterol. Clear? Though your options are hyperthyroidism, nephrotic syndrome, hypothyroidism or obstructive jaundice. Basically, nephrotic syndrome, hypothyroidism, obstructive jaundice. These all are wrong options because in hyperthyroidism, the secretion of T3 and T4 increases. Clear? And with the increment of these two hormones, T3 and T4, T3 is thyroxine, T3 is tridothyronine, and T4 is thyroxine. Your metabolic process also increases. Clear? So, when your metabolic process increases, cholesterol metabolize. Clear? Cholesterol metabolize. and it does not accumulate in your blood. So obviously hyperthyroidism is that condition in which hypercholesterolemia is not found. Move on to the next question. The following are the ketone bodies except acetoacetyl coenzyme A, beta hydroxybutyrate, acetone or acetoacetate. Basically, our body contains three ketone bodies which get stored in your liver and also forms in liver with the help of ketogenesis, as I said you before. So the correct option will be acetoacetyl coenzyme A, which act as a intermediate molecule in the process of ketogenesis. Basically, when you combine two acetyl coenzyme A, they forms acetoacetyl coenzyme A. Acetoacetyl coenzyme A. So basically, it is a type of intermediate molecule. Intermediate molecule. Clear? So the correct option will be A, acetoacetyl coenzyme A, because beta hydroxybutyrate, acetone and acetoacetate are the ketone bodies which found in human body. Next question. The insufficient exposure to sunlight may cause rickets in children because now, as you all know, that sunlight is the major source of vitamin D, that is called calciferol. And if it gets deficient in children or adults, it causes rickets in children and osteomalacia in adults. So you have to tell the reason behind this. Sunlight has magical powers? No. Sunlight contains vitamin D? Wrong. Sunlight induced synthesis of vitamin D3 in the skin is decreased, causing vitamin D deficiency. So basically, option number C will be the correct option. Basically, sunlight does not contain vitamin D. So what is the reason? Your body have cholesterol which gets converted into vitamin D on the exposure of sunlight. Clear? So the correct option will be C. Sunlight induced synthesis of vitamin D3 in skin is decreased, causing vitamin D deficiency, that is rickets and osteomalacia. Osteomalacia. Clear? Next question. Increased prothrombin time is observed in the deficiency of vitamin K, vitamin B, vitamin A, and vitamin B12. What is prothrombin time? Prothrombin time is a type of test. Evaluated test, which used to evaluate the blood clotting time. Basically, vitamin K is that vitamin whose deficiency causes increment in prothrombin time test. So the correct option will be vitamin K, which is also known as phyloquinone, phyloquinone, and also coagulation factor, factor. Clear? Next question is, the enzyme catalase destroys hydrogen peroxide by forming hydrogen and oxygen, water and molecular oxygen, water and nascent oxygen, or all of these three. Basically, what is hydrogen peroxide? H2O2. When it gets hydrolyzed with the help of catalase, it forms water and oxygen, which is molecular oxygen. Clear? So the correct option will be second, water and molecular oxygen. This is hydrogen peroxide. With the help of catalase, it gets converted into H2O and one molecular oxygen. So the correct option will be second, that is water and molecular oxygen. Move on to the next question. Which of the following vitamin is involved in ETC, that is electron transport chain? Electron transport chain. Your options are thiamine, that is vitamin B1, folic acid, that is vitamin B9, riboflavin, that is vitamin B2, and cobalamin, that is vitamin B12. Basically, riboflavin, that is vitamin B2, is responsible, or you can say is included in electron transport chain. But how? Riboflavin forms coenzyme, which are also known as FAD or FADH or FADP. FADP. So these are the coenzymes which are involved in electron transport chain, and these coenzymes are formed with the help of vitamin B2, that is also known as riboflavin. So the correct option will be C, that is riboflavin. Next question. The log and key theory of Fisher states that, now you all have heard about this log and key theory of enzymes. Your options are: active site in enzyme exists in proper conformation of the substrate molecule even in the absence of substrate. No, this is not the correct option. Active site undergoes conformational change. Stop here. Wrong. There is absolute specificity in reaction between enzyme and substrate. This will be your correct option. But how? Let's draw an example. Basically, see. For example, this is your enzyme. This is active sites, and it is your substrate. As you all can see, active site of an enzyme and substrate has a proper conformational affinity, so that this substrate gets bind with this active site of enzyme properly and forms enzyme substrate complex and then enzyme product complex. Clear? So the correct option will be, there is an absolute specificity in reaction between enzyme and substrates. Next question is, allosteric modulator influences enzymatic activity by enzymatic activity or allosteric modulator. What is allosteric modulator? That is allosteric inhibition. Clear? Competition for active site with substrate. It is not the correct option because competition occurs in competitive inhibition. And the question is asking about allosteric inhibition. Second is binding to the site on enzyme distinct from active site. What is the meaning of distinct? Other than active site. Clear? So the correct option will be second, obviously. Binding to site on enzyme other than the active site. This is allosteric inhibition. Clear? And how? This is how it influences enzymatic activity. So the correct option will be second. Next question is, which of the following enzyme is not involved in gluconeogenesis? What is gluconeogenesis? Gluconeogenesis is the process by which new glucose synthesize. New glucose form or synthesize. Clear? Your options are pyruvate carboxylase, phosphoenol pyruvate, carboxycinase or hexokinase. Basically, correct option will be hexokinase because it is an enzyme. It is an enzyme of glycolysis. Clear? So the correct option will be D, that is hexokinase, which is an enzyme of glycolysis. Clear? Next question, the nucleic acid base found in mRNA but not in DNA is adenine, cytosine, guanine and uracil. Basically, our body contains 5 types of nitrogenous bases, that is adenine, cytosine, guanine, uracil and thymine. Clear? Thymine. Out of which uracil is that nitrogenous base that is only found in RNA. Either it is mRNA, tRNA or rRNA. So the correct option will be obviously uracil. Clear? Next question. Double helical structure model of DNA was proposed by Pauling and Corey, Peter Michel, Watson and Crick, King and Wooten. So the correct option will be obviously Watson and Crick. That is James Watson and Francis Crick. James Watson and Francis Crick. Clear? So they propose the double helical structure model of DNA. There are many postulates about this double helical structure model of DNA. So you should know about those postulates. Next, second last question is, starch consists of, what is starch? Starch is polysaccharide. And which type of polysaccharide? Homopolysaccharide. Homopolysaccharide. What is the meaning of homo? Homo means a polysaccharide which contains only a single type of monosaccharide unit, that is glucose. If you talk about starch, it contains only glucose monosaccharide units. Basically, the forms of glucose is occurs in two types like amylose and amylopectin, from which amylose is a linear structure, and amylopectin is a branched structure. Clear? So obviously the correct option will be second, unbranched amylose, or you can say linear amylose and branched amylopectin. These are the forms of glucose in starch. Clear? So the correct option will be unbranched amylose and branched amylopectin, which are connected by glycosidic linkage. Glycosidic linkage. Next and last question, which of the following glycosidic linkage is found in maltose? Now what is maltose? Maltose is disaccharide. Disaccharide. Clear? Glucose alpha 1 to beta fructose, glucose alpha 1 4 glucose, galactose beta 1 4 glucose, glucose beta 1 4 glucose. Before coming to the options, you should know about the monosaccharide units which are found in maltose, that is 2 glucose units. So obviously the correct option will be B or D. Clear? Basically, the glycosidic linkage which is found in maltose, that is alpha 1 4 glycosidic linkage. So the correct option will be second, glucose alpha 1 4 glucose. Clear? So these are the best 25 MCQs questions of biochemistry which are very important for your upcoming Telangana pharmacist exam. For PDF, you can join Telegram. Link is in description box. See you in the next video. Till then, stay tuned with Pharmacy India. Thank you.